So you can take these points on a map and reconstruct the range of a species. And we've talked before about the potential for sometimes you get points on a map that are not exactly natural points. So sometimes there's a mistake in how the data point was entered, or sometimes you have an actual collection of a plant that's just outside of its native range. So we talked about native and non-native plants before. A lot of times you can piece it together based on the specimen information. So if you have a specimen of this plant that's collected in a garden, so it says it's from somebody's garden or a horticultural center or a botanical space, that might be outside of its native range. So this is the plant that people get quinoa from, the edible, they call it a pseudo grain. And it's collected from South America, quite a few collections, a whole bunch from United Kingdom, Europe, a few from North America, Central America. So where do we draw the lines on this map to say that this species is native? So to determine whether plants are native, you can actually use the specimens, and really the first specimens that are collected from an area before, we're usually talking about people of European heritage, before those people were spreading their culture and moving plants around. That was kind of the snapshot of when things were native. So if you have specimens that are collected after that time, you might have reason to think that they were grown in cultivation. And all that information can be kind of compiled together. So you have data from the original discoveries, discoveries in quotes, people from Europe coming to so-called discover plants in North America, South America. What they found at that time is generally regarded as native, but after that time, sometimes they moved around. So one of these resources, this is from the Plants of the World online resource from the Kew Botanical Garden. They have something that was really missing up until a few years ago. Nice resource for being able to determine where plants are native or not native. So on their maps, green is native, purple is non-native. And so this plant has been determined to be native to the Andes mountain range. People knew that basically. So I didn't show you the rest of the map, but this is the only place the species is native. So growing in North America is outside of its native range, probably in cultivation. Growing in Europe is outside of its native range. So, they have a nice resource online that will tell you what's native and non-native, but this is based on specimen data as far as when those specimens were collected, and maybe a little inference about how they got to be where they were at the time of the collection. Well, you could take these data and compile them over time. So this is actually a little graphic that I put together for this species. This is Japanese knotweed, which is a non-native species in North America. And I went through and just took data from the GBIF database. And for every 10 years, I built a cumulative map of where all the specimens had been collected up to that point. And you can see a pretty good reconstruction of this plant showing up on the East Coast. No surprise there, that's an area of high population density. And then moving from that area, either because people are moving them around or because they're able to move around biologically on their own. But this is an important part if you're working with an invasive species or a non-native species, an important step in kind of putting that together and saying, okay, well, where did it come from? When did it get to this area? Where's it going next? Things like that. So here's Japanese knotweed, another one. Another invasive species, this is Hydrilla, a water plant, has a little bit different story coming in from more southern location and filling in a lot faster. So this is one of the more notorious aquatic invaders, Hydrilla, and you can see it filling in pretty nicely there. So again, every point on here is a specimen, and they go back in time to really the first time when anybody ever collected this plant in North America. And I don't know how many total, quite a few specimens by the end of this. So when we look at invasive plants, we're often thinking about them expanding their range over time. If we're looking at native plants, sometimes native plants actually lose their range over time. So I think a nice perspective to take for the state of Wisconsin, and of course much of North America, would be how are these plants different at the time when people of European heritage were first moving across this area. That's the map on the left. This is called, they call it pre-settlement vegetation. Basically, the first people who came through, some of the first people coming from the East Coast were surveyors that came and surveyed the land as far as what species are there, what kind of ecological communities were present. And so this map indicates that information. And we've got different forests and prairies and so on. And shows our nice tension zone across the middle. And that was the pre-settlement vegetation. And now we're living in the decidedly settled time. So this pink magenta color here is urban areas. These are our bigger cities. And then just this giant swath of yellow. So we live in a very agricultural part of the country, as you probably know. And all this area that's now agricultural, there's just field after field of managed cropland. And so those crops, they grow in an area that's easy to grow crops, but there were natural communities in those places before the crops came in. So a lot of times the communities that were there before settlement, certain types of them are really at risk because they were the ones growing on this more commercially desirable land. So these deciduous forests over here, a lot of these are gone. The area where this was on the left map is now just about all yellow on the right-hand map. So some species end up becoming endangered in those areas and then potentially over time even lost. Just zooming out a little bit more, I kind of like this one, just showing a loss of forests. So again, as European settlers came through, they were taking over space, turning it into farmland to support themselves and to make money off of farming areas. And they basically cleared the lands that could be cleared. So a lot of this southern oak forest and savanna habitat, this kind of brown, light brown color, a lot of that was cleared out. You can see here, this is a lot of the driftless area that we talked about previously. Driftless area, not so great for cultivating. So left intact because it's may have these kind of habitats, but just not really good for turning into farmland. Other examples, this is one relatively recent study from Minnesota, just highlighting, again, using specimens, going through time and just noticing how these ranges are decreasing. They were surveying the plants found in lakes currently relative to what has been recorded as being there in the past. Quite a few of these red dots are indicating loss of plant diversity. So basically species that were known to grow in these lakes that are no longer present in those lakes. So loss of diversity in those areas. Not only that, but some of these are actually described as drained lakes. So if you're an aquatic plant and your lake has been drained, that's kind of the end of your local habitat. So really good to have this record as a historical record. It's really hard to go back in time and, have anything written down about what plant species were present in a location. But if you go right to the specimens, you can get that information. As I mentioned before, you can record the phenology over time. We haven't talked about phenology. Phenology is the study or the expression of what stage of their life cycle plants are going through at any one time. So phenology talks about like what time of year is the species flowering? What time of the year does it first have its leaves in the spring? What time of year does it produce fruits and so on? What time of year do leaves change color and fall off of the trees? And using specimen data, this has become the basis for understanding a lot of climate change. So we have the non-biological climate change where we can have records of temperatures changing, precipitation changing over time. But the biological record, a lot of it comes down to something like this, where we notice that plants are leafing out at different times of the year, producing flowers at different times of the year. So if the earth is getting warmer, or if a local habitat is getting warmer, a lot of plants are keying into temperature as their cue for when to start flowering. So if things are warmer, if conditions are warmer, you get earlier flowering. And this is what's highlighted in this illustration. This is a study from the Northeast of the United States. It takes advantage of some pretty thorough data that Henry David Thoreau actually collected. He was a naturalist that you've probably heard of around that area. Collected data about when his plants in that area, plants that he knew about, when they were first flowering every year. And from his location in 1850s up until 1890s, a different person, and then now again in the recent time, 2000s, people are checking these days when these flowers are blooming, and the trend line, if you connect these dots, the trend line is decidedly earlier. So we're going from 15th of May to the 5th of May for the same species to produce their flowers. So 10 days a year on average, pretty significant change in plant species. And this raises concerns about who's going to pollinate them, whether that upsets the pollination systems. Another concern with maybe species changing ranges as different parts of their habitat become warmer or colder. You can use specimens, as I mentioned before, to kind of reconstruct ecology. One method that's used is it's basically a computer modeling mechanism, technique, where you take the locations where a particular species has been found and you try to extract or extrapolate from their locations what particular habitat tolerances they have. Are they more tolerant to soil types or temperatures, maximum temperatures, minimum precipitation, other plant species abundance, and so on. And so this is an example of a plant from Brazil, showing a few places where it has been collected, and then the colors on the map are indicating basically the likelihood that that is a suitable habitat for this plant. So the red is a highly suitable habitat, a couple dots a little farther outside of that, supposedly suitable habitat. But this is a nice perspective to have when it comes to maybe conservation. If you have a plant that's getting rare and you identify the places where it potentially could grow, that gives you potentially options to introduce it somewhere, or habitats that you want to target as far as ones to conserve. I mentioned this before, climate change. So changes in the global temperatures are changing the areas where different plant species can grow. So if you model where a plant is able to grow and that's dependent on temperature, you might have a different place where it's able to grow after climate change. So this one's mapping global radiation over time, the past, from 1994 to projected to 2050, big change in global radiation and potentially changing the habitats where these plant species can grow. Other uses for plant specimen data, you can mine them for information about their morphology. So this was a study actually that I did, and I have these two species in North America, the east of North America, two species in the genus Nymphoides that are mostly easy to tell apart, but sometimes they have individual plants that are a little bit difficult to tell one from the other. So I undertook a study to do a lot of measurements using herbarium data, actually using electronic versions of herbarium data. So a lot of times like a scan of a specimen, and taking measurements of these and determining what the range of variation was, even across its entire range. What's shown here are these two different species, one in dark blue and one in brown, showing two of the example measurements, and how they are mostly different, but with a little bit of overlap in here. So stem diameter, mostly short in the blue species, mostly long, or mostly longer in the brown species, but not always independent from each other. So this is the area, these dotted lines are individual specimens that are harder to identify. So same thing with the lamina, this is the leaf blade width. Some of these individuals difficult to tell apart because they're in this intermediate range. Some individuals pretty cleanly identified as one species or the other. But this was basically applying quantitative morphological data to try and make this distinction a little bit more clearly. So making those distinctions, this becomes the basis for writing out a dichotomous key, as we have seen. So when you see these ranges, up to this point, we've been using other people's ranges, but you could develop your own range of measurements using a collection of specimens and determine that one species has a particular range of measurements and another species has another range of measurements. And then that becomes the basis for your dichotomous key. So seeing that the diameter of the involucre is two non-overlapping ranges. That could be the basis for probably a very important distinction here. Florets here, we got between 3 and 13. Here we got between 10 and 60. So mostly non-overlapping range there. A person would have to look at a whole lot of specimens to be able to determine these kinds of things. We talked previously about describing new species. Just a reminder that if you need to know that your species is different from any other species that's already been described, you need to be able to compare it to those other species. And mostly, a lot of the times, if botanists are doing any kind of measurements or determinations about variation within a species, they're not going out into the field. I mean, a lot of times you'd be looking at huge areas of land that you would have to cover to collect representative specimens of a species. How much better is it to just go to a herbarium and collect a bunch of data all at once, all in one place? So if you've looked at representative specimens across the range and you find out that you have a species that is outside of the range of those other species, that can be one of your strongest pieces of evidence for saying that you have a new species. These days, we talked a bit about herbarium, or about DNA and phylogenies. This is just a massively growing pursuit of people trying to reconstruct phylogenies using DNA. We have new perspectives on how species are related to each other. And more, if you have species that you need to investigate, and that species is some other grows in some other part of the world, maybe one that's difficult to get to, how much easier than that would be just to grab specimens from your herbarium. So fortunately, if you have a good quality specimen, you can get lots and lots of DNA, even out of a dried specimen that's been dried for 100 years. I don't know what the record is now, but people can get usable DNA from very old specimens these days. And then they turn that into new evidence about the particular question that involves DNA or phylogenies. Including this one was kind of interesting from not too long ago, investigation of cultivated potatoes. So the potato that ended up in Europe and then back around to North America, that was originally a plant that was native to South America. So potatoes are native to South America, and became this question that this paper addressed of which part of South America. So there were in Europe, after they'd come back to Europe from South America, these potatoes had shown up, and they had some from the northern Andes and some from Chile. And they basically connected those to when those specimens were collected and which one was more related to the cultivated potato. So a combination of specimen data and DNA data to answer that question.